Hello, in this presentation I'm gonna talk about antibiotic resistance. It's a topic related to pharmaceutical microbiology. We are gonna discuss the main causes of the origins of the resistance and also the mechanisms that bacteria use to create this resistance against the main antibiotic that we use. Antibiotic resistance is the ability of the microorganism to inactivate or to negate the harmful effects of the antibiotic. What does that mean? It means that the microorganism survives even though it's under attack of an antibiotic. So it creates resistance against the attack of that antibiotic. It has increased a lot during the antibiotic era because if you are using antibiotics more and more bacteria will get resistant. And this is the main cause is because of mutual competition in the environment between the organisms that produce antibiotics. So if I have a population of bacteria that produce antibiotics, they compete between each other by resource or whatever, and then they create resistance against that antibiotic. It's like a natural selection. The main types of resistance are multiple drug resistance, MDR, is when bacteria are resistant to at least 3 different classes of antibiotics. And the other one is pan-resistance, is when bacteria is resistant to all non-antibiotic classes in use. So the multiple drug resistance is not so dangerous as the pan resistance because in the pan resistant bacteria are resistant to all all antibiotics in use. The research in new antibiotics has decreased a lot in the past decade. So as you can see from 98 to 2010 it has decreased almost 3 times. So research in the development of new antibiotics has dropped in the last 20 years. It's because it's not so profitable to the companies. That's why they just stopped to develop new antibiotics. And so even worse is that there are even fewer options to treat resistant bacteria. And the resistance usually develops in 10 or 15 years, but some resistance has been reported in just 1 year. So bacteria after the development of a new antibiotic, bacteria got resistant in just 1 year. So here is some examples about when the antibiotic has been created and when the resistant bacteria has been reported. So for example, the penicillin, the first antibiotic introduced in 1940s, the first resistant bacteria was recorded in 1965, so about 22 years after the creation of the antibiotic. For tetracycline, it was faster, so the first resistant bacteria was reported in just 9 years. For methicillin, just 2 years. For ceftazidime, in just 2 years also. And this is antibiotic that is the third generation of cephalosporin. For levofloxacin, it had resistance in the same year, so it was created in 1996, and in the same year they recorded some bacteria that was resistant. For linezolid, it's just 1 year also. This is an antibiotic that's protein synthesis inhibitor. Daptomycin is the only antibiotic that has not been detected resistance yet. This is a cell membrane disruptor, but it's the only one with no bacterial resistance yet. Observe the resistance. Bacteria get resistant much faster nowadays. The major candidates, multiple drug resistant candidates, so MDR, are 3 main groups. The first one is the urgent group. So this is an immediate public health threat because it requires urgent and aggressive action. For example, Clostridium difficile, carbapenem resistant Enterobacteriaceae, and drug resistant Neisseria gonorrhoeae. We are going to talk about them later. The other group is the gram-positive. The main gram-positive that are MDR are Staphylococcus aureus, Enterococcus, Streptococcus pneumoniae, and Mycobacterium tuberculosis. And for gram-negative, the main species are Actinobacter baumannii, Pseudomonas aeruginosa, and Salmonella. So first I'm gonna talk about the urgent. So the urgent are the most hard bacteria to be combat. So for example, Clostridium difficile, we have 25,000 infections per year and about 14,000 deaths, with more than 1 billion costs in medical treatment. So Clostridium difficile is a life-threatening bacteria because it causes life-threatening diarrhea. It's commonly hospital acquired. It's treatable, but the spread of the resistance is very fast. So we can treat this bacteria, but their resistance spreads very fast. The 400% increase in health outcomes occurs in between 2000 and 2007. And 50% of the infections occur in people less than 65 years old. So a half of percent of the total of infections is with people with 65 years old or less. And the other half is older people, like more than 65 years old. Another species is carbapenem-resistant Enterobacteriaceae. It causes about 9,000 drug infections per year, this resistant bacteria, and 600 deaths. It also has become resistant to all or nearly all available antibiotics, so it's very hard to treat them. The main bacteria is Klebsiella and Escherichia. And 50% of people who are contaminated die, so it's a high level of death. There is at least one type of carbapenem-resistant Enterobacteriaceae in every healthcare facility in 44 states of USA. So in USA, we have at least one type of this bacteria for in each hospital or healthcare facility in USA. For Neisseria gonorrhea, it causes around 246,000 infections per year. It's the second most common infection in USA, and the infections affect the reproductive organs and the urinary tract, and it can make complications in the reproductive system. So it causes problems for the reproduction. Now I'm gonna talk about the gram-positive. So the first one is methicillin-resistant Staphylococcus aureus. So Staphylococcus aureus that are resistant of methicillin is one of the most dangerous bacteria, gram-positive. Another one is Enterococcus that are resistant to vancomycin. This resistance was found in genes of ancient bacteria. What does that mean? Is that this bacteria has developed this resistance a long time ago. So ancient bacteria that have this gene against this antibiotic. And penicillin-resistant Streptococcus pneumoniae, it is especially important in developing countries. So in developing countries, this is a very dangerous bacteria resistant against penicillin. And Mycobacterium tuberculosis is also another one that can cause death. Now related to gram-negative, I have Actinobacter baumannii. The main characteristic of these bacteria is that they have big plasmid with several resistant genes. So if they have big plasmid, they can carry more and more genes. So it's easier for this bacteria to get resistant to multiple drugs. Pseudomonas aeruginosa is also another one that is main found in soil, for example. And Salmonella species is especially important in developing countries, especially if we don't have like treated water or any care about the food or the water. Now I'm gonna talk about the origins of resistance. So the first one is innate resistance. Is like when the bacteria has this resistance by itself. So they develop some variations in the structure of the cell envelope, for example, decreasing the permeability of the antibiotics. So they have, for example, efflux pumps and purines, which reduce the cell uptake. So the flux pumps, they pump out the antibiotics, and the purines pump in. But if bacteria decrease the number of purines, it's gonna take less antibiotics. We're gonna talk about them later. So here, for example, the purine is from where the antibiotic got in. So if the bacteria has less purine, the antibiotic is gonna have less space to go into the bacteria. So bacteria who has less purine is gonna have less, it's gonna be less affected by the antibiotic. And another origin of resistance is phenotypic, is when bacteria adapt to grow and to survive in an environment with antibiotic content. So when the bacteria is exposed to antibiotic, it develops some resistance. For example, if the bacteria has a plasmid that contains the gene against that antibiotic, the bacteria is gonna keep that plasmid. But if the bacteria is in an environment that doesn't have the antibiotic, then the bacteria is gonna lose that plasmid because it's not useful, and the plasmid requires energy to be kept. So, if, for example, a bacteria is in a media with ampicillin, the bacteria is gonna keep the plasmid. But if the bacteria is living in a media without ampicillin, bacteria is gonna lose that plasmid because to keep that plasmid require energy. So bacteria is gonna keep this plasmid only if the bacteria is in an environment that has the antibiotic. Another phenotype is the conjugative plasmids, also called R factor. What's that? Conjugative plasmid or R factor is a plasmid that has multiple resistant genes. So it carries resistance against different classes of antibiotics and other microbials under the same promoter. So what's important about it? So if I have a plasmid here that has this promoter and I expose the bacteria to ampicillin, for example. If the bacteria has ampicillin in the environment, it's gonna activate the beta-lactam resistance. So the promoter is gonna be activated to to activate the genes for beta-lactam resistance. But under the same promoter, the bacteria also have genes against tetracycline, chlora, chloramphenicol, streptomycin, and sulfonamide. So if I activate beta-lactam resistance, I'm gonna activate all the genes also. So bacteria will get resistant not only against ampicillin, but against all these antibiotics because they are under the same promoter. So if I activate one of them, all of the others is gonna be activated as well. Some examples of bacteria that has R factors are Pseudomonas aeruginosa, RP4. So this plasmid has genes against ampicillin, kanamycin, and tetracycline. R1 in gram-negative, so the genes has ampicillin, sulfonamide, and trimethoprim. And PSH6 in Staphylococcus aureus, it has genes against gentamicin, trimethoprim, and kanamycin. Bacteria will contain this resistant gene. Gene or horizontal gene transfer is when one bacteria passes the plasmid to another bacteria that are not directly related. For example, it's not daughter cells to it's not mother cells to daughter cells, but bacteria that pass to another bacteria that are not her daughter cells. This is horizontal gene transfer. Another phenotypic origins of resistance is the insertion of sequence or transposons. Transposons are genes that are mobile, so they can move in the genome to another location. So here is an example. I have this uh, insertion sequence. In my insertion sequence, I have the transposase genes. What's that? Is the genes that can be mobile. And I also have the inverted repeat sequence that are recognized by the transposase. So if I don't have this inverted sequence, my gene is not able to move to another location. And here I just have the transposase genes. Each gene has the antibiotic resistant genes and the essential genes for this movement in the genome. And move from here to another location. So when I move them, they may be activating genes of resistance. Another phenotypic origin of resistance is the mutation. So mutation is when bacteria create new genes or new sequence that may have resistance against some antibiotics. So for example, if I change a sequence, I may replace my amino acids and I may create new proteins or the enzyme that's gonna have different function, and one of these functions may be the resistance of against an antibiotic. So mutation is another phenotypic origins of resistance. Some considerations regarding the origin of resistance is like resistance is the collection of all the antibiotic resistant genes in both pathogen and non-pathogenic bacteria. So the resistance may be recorded to any bacteria, even pathogenic or non-pathogenic. So any bacteria can get resistance against antibiotic. Even though the bacteria doesn't cause disease, the bacteria can have these resistance genes. These antibiotic resistant genes are very common in soil antibiotic producers because soil is an environment that has a lot of different species of bacteria, so they compete between each other, so they develop resistant genes very easily. The antibiotic area has increased immensely the environment selective pressure. So in the soil, it's like a natural selective pressure, but when we create new antibiotics, we introduce new pressure for bacteria to create resistance. So with antibiotic area that starts from our 1940s, we increased a lot the antibiotic resistance. This is mainly caused because of medical practice, so to treat disease, but also to veterinary and agriculture use, for example, by the use of feed additives for animals or plants. Also in food industry, for example, when a company use antifungal agents and preservatives to preserve the the spoil of the food, it also creates a selective pressure in that population of bacteria. And other one that is not so controlled is the household products. So, for example, triclosan, soap, sportwear, sanitizer, and the excessive use of disinfectant. This is the main reason and source for bacteria resistance because for these products there is no strict regulation, so everyone can use for free, so they can use as much as they want. So this creates a higher diversity of class of resistant bacteria. So this is not controlled by any regulation, so this is the main source of bacterial resistance. So Now I'm gonna talk about how the bacteria is spread. So this is very common sense. For example, a guy has a disease, it goes to the hospital, and in the hospital it may be in contact to other bacteria and other personal, um, for example, other people who are there in the hospital to be treated, or even the people who work there, nurses, doctors, so they are in contact to that bacteria. And then they go back home and they spread the bacteria. Or by an animal, when I have the contact or when I eat an animal, I may have um, take some bacteria from them. Okay, so even not just animal, but some plants also from farms, we also can get. So everybody is connected and then spread the bacteria very easy. Now I'm gonna talk how the main mechanisms of resistance against antibiotics that bacteria use. So we have four major groups. The first one is the enzymatic activation of the drug. So what does that mean? Is like the bacteria breaks down the antibiotic enzymatically. So if the bacteria breaks down the antibiotic, then the antibiotic is not gonna make its function properly. Example, beta-lactamase. So this antibiotic, bacteria use this enzyme to break down the antibiotic beta-lactam. Another group is the alteration of the drug target. So bacteria change their conformation and the antibiotic cannot bind anymore. So for example, if antibiotic attacks a ribosome, so what bacteria does is that bacteria change the format, the shape of that ribosomes, for example, by adding methyl groups, and then the antibiotic cannot recognize that ribosomes anymore, and so the antibiotic cannot attack. Another one is the penicillin bind proteins protection. So PBB is the main uh, is the is the is the mm, is the little piece that form the cell of the bacteria. So penicillin, for example, acts against this precursor of cell wall. And then bacteria change the shape of this PBB. If the bacteria change the shape, the antibiotic cannot recognize anymore. Another group is the reduced cellular uptake, is when bacteria change their cell membrane by decreasing the number of pores. So bacteria has less spot for the antibiotic to get in. And finally, the fourth one is the increased flux of the drug. So bacteria uh, is constantly pumping out the antibiotic away from the cell. So antibiotic goes in, but bacteria has the flux pumps to pump them out. And the clinical relevance is from 1 to 4. So 1 is the most clinical relevance. So what does that mean? Is that this one is more important. We need to be more careful about this resistance than the increased efflux of the drug. Okay, so enzymatic activation of the drug is more of concern than alteration of the drug target, then the reduced cellular uptake, and then increased efflux of the drug. Now I'm gonna talk about each of them. First, I'm gonna talk about the enzymatic activation of the drug. So as I said, the bacteria cleaves the antibiotic, so the antibiotic has no prop property to act against the bacteria anymore. So here's an example. Here I have my penicillin, and here is my beta-lactam ring in the penicillin. So what beta-lactamase enzyme does, it breaks down this beta-lactam ring. So if this beta-lactam ring is broken, then the antibiotic is not active anymore. So this is an enzymatic activation of the drug. This predominantly affects natural antibiotics. So Penicillin, cephalosporin, and carbapenem are natural antibiotics that bacteria has enzymes to break them down. And these resistant genes can be encoded in both plasmid and genome. Okay, and this resistance is higher in gram-negative than in gram-positive. So this gram-negative makes this more often than gram-positive. So how to fight the beta-lactam resistance? The first way to fight this resistance is to use semi-synthetic modification of the antibiotic, for example, methicillin. So semi-synthetic antibiotics are better because bacteria doesn't have resistance for so long. So these are kind of new antibiotics, so bacteria didn't have enough time to create a natural resistance. Another way to fight this is to combine this antibiotic with beta-lactamase inhibitor. So, introducing the antibiotic a chemical that has an inhibitor of the enzyme. So, this chemical inhibits the action of the beta-lactamase enzyme. But this chemical has no antibiotic activity itself, okay? So it's just to inhibit the activity of the enzyme of the bacteria. One example is augmentin. It's a combination of amoxicillin, that is a beta-lactam antibiotic, and clavulanic acid. This clavulanic acid is the beta-lactamase inhibitor. The benefits of this drug is that this drug does not kill the bacteria because the bacteria does not. So this antibiotic does not kill the bacteria. This chemical clavulanic acid does not kill the bacteria. So bacteria does not recognize this chemical as a problem. So if the bacteria does not recognize this as a problem, bacteria will not create resistance. So it's a way to slow down the resistance of the bacteria. So this gives a new life to the existing antibiotics. This is faster and cheaper. However, some bacteria already have resistance against the beta-lactamase inhibitor. So some bacteria already developed the resistance against the inhibitor. One example is the metalloenzymes, is enzymes that has metals as a cofactor that decompose the inhibitor. So even though this way is better to fight the bacteria, but some has already developed this resistance by the use of metalloenzymes. Now I'm going to talk about the alteration of the drug target. So what's that? Bacteria change their conformation, the conformation of the target, and the antibiotic cannot bind anymore. So for example, ribosome protection. So let's say antibiotic is acting against a ribosome. So what bacteria do is bacteria add methyl groups in that ribosome. So it creates like a shield um, around the ribosomes, and then the antibiotic does not recognize anymore. Okay, so here I have the RNA, and bacteria add methyl group. This is called methylation. And then if my ribosome has methyl groups, so my antibiotic does not recognize anymore. So this is the alteration of the drug target. So the target of the antibiotic has changed, not the antibiotic. Another example is the penicillin bind proteins protection. So bacteria, as I said, PBP is the precursor of the cell wall, and the penicillin acts against this precursor. So if the antibiotic fits exactly here, then the antibiotic is gonna break down this PBB and the cell wall will not be formed. But if the bacteria change the shape of this PB, the antibiotic will not be able to bind, so it's not gonna be affected. Okay, so if bacteria change the shape of PBB, the antibiotic will not be able to bind, and there won't be any inhibition on the cell wall synthesis. So the bacteria will survive. This is higher in gram-positive than in gram-negative, and it's most relevant in Streptococcus pneumoniae that cause pneumonia, medicine-resistant Staphylococcus aureus that cause bacteremia, and Haemophilus influenza that cause meningitis. So these three main bacteria have this kind of resistance. Now I'm going to talk about reduced cellular uptake. This is when bacteria reduce the number of proteins that it has. So, as I said before, if bacteria has a lot of proteins, it's going to have a lot of room for the antibiotic to go in the bacteria. But if the bacteria reduces the number of proteins, it's going to have less space for the antibiotic to go in. So, if I decrease the number of proteins, I reduce my antibiotic uptake and I increase my resistance. And the last one is the increased efflux of the drug. So efflux pumps are protein complex, as you can see here. So they are proteins that they recognize the antibiotics that are in the cytosol and pump them out from the cell. Okay, so the efflux pumps reduce the effective concentration of the antibiotic, but this is only of clinical relevance if it's overexpressed. So if I have a lot of efflux pumps, it's gonna be a concern. But if it's like not so much, it's not of a clinical relevance. So if I increase my number of efflux pumps, I decrease my antibiotic uptake and I increase my resistance. So just to clarify proteins and efflux pumps, both of them are protein complex that form both of them are protein and they are formed by upregulation of the gene. What does that mean? They are regulated by the genome, not plasmid. So they require the expression of gene, mRNA, and protein. Okay. If they are encoded by the genome, not plasmid, they can only be transmitted through replication of DNA. What does that mean? By vertical gene transfer. That is from mother cells to daughter cells. So cannot be passed to different species or species that are not daughter cells. So they are also very stable. So because plasmid can be easily lost or bacteria can stop produce them, but genome no. The genome are stable. Bacteria cannot just lose the genome. Okay, so if bacteria cannot lose the genome, the gene that produces proteins and the flux pumps will not be lost. Okay, now I'm gonna talk about specific groups of antibiotics. So the first one is the glycopeptides. So glycopeptides, they are also known as vancomycin, are are antibiotics that inhibit bacterial cell wall formation by inhibiting peptidoglycan synthesis. So what does that do? So here, for example, I have the precursor of the cell wall. They need to be connected by the transpeptidase enzyme to form the cell wall. But what the glycopeptide does, they bind to this precursor, and then this precursor cannot form the cell wall. Okay, so they bind to cell wall precursor, not the enzymes. Okay, they don't bind to the enzymes, they bind to the cell wall precursor. If the cell wall precursor cannot be linked, the cell wall will not be formed. Now, how bacteria get resistant against glycopeptide? So what they do, they change the format of the cell wall precursor. So the cell wall for a susceptible cell, so a normal cell, The cell wall precursor has the alanine terminal. If the cell wall precursor has the alanine terminal, the glycopeptide can bind regularly, and the cell wall synthesis will be inhibited. But if the bacteria change the cell wall precursor from deaniline terminal to D-serine terminal, then the glycopeptide will not get affinity to that precursor, so will not be able to bind, and then the cell wall synthesis will not be inhibited. So the precursors will continue to do their job without interference of the antibiotic. Okay, now I'm going to talk about aminoglycosides. First, I'm going to explain the mechanism of action. Okay, so aminoglycosides, they bind to the ribosome, interfering with the protein synthesis. So what they do, they bind to the ribosome. So here they bind to the ribosome. If they bind to the ribosome, it's gonna cause mismatch of the codon. So when the tRNA binds here, it's not gonna bind exactly when it's supposed to be bind. So they're gonna bring the wrong amino acid and they're gonna form the wrong protein. Now, how bacteria got resistance? First, bacterial enzymes modify the drug, reduce the drug's ability to interact with the ribosome. So, bacteria attacks the drug and change the format of the drug. If the drug is changed, it cannot bind to the ribosome anymore. Okay, so the main cause of resistance is when bacteria change the format of the antibiotic. So bacteria directly attacks and modify the drug. There are two enzymes that attacks the drug. One of them is aminoglycoside kinase that add phosphates to aminoglycoside. So as you can see here, for example, I have the canamycin antibiotic. Bacteria adds phosphate groups to that antibiotics. So if I add phosphate groups, the antibiotic will not be able to bind to the ribosome anymore. Okay, the enzyme that does that is the aminoglycoside kinase. And another enzyme is the aminoglycoside acetyltransferase. Instead of adding phosphate, it adds acetyl group to the aminoglycoside. So here I have the antibiotic, and here I have the antibiotic that bacteria adds the acetyl group. So the same way, if the antibiotic has this acetyl group, it cannot be able to bind to the ribosome anymore. Okay, so if the bacteria does the same, change the antibiotic, but add in different groups. So the enzyme kinase adds phosphate, and the enzyme acetyltransferase adds acetyl group. Another way, they also have negative charge with repel RNA and DNA. So this enzyme has negative charge that also repel RNA and DNA. So this negative charge here of the phosphate and the acetyl group, they repels RNA or DNA because RNA and DNA also have negative charge. Okay, so my phosphate group and my acetyl group has negative charge that repels RNA and DNA. Another way to get aminoglycoside resistance is through ribosome methylation. That's already said. It's just adding methyl groups to my ribosome. Okay, so if I add methyl groups to my ribosome, the antibiotic cannot be connected to them anymore. They are exhibited by aminoglycoside producing organisms. They are of little clinical relevance except when I'm using macrolides. Okay, now I'm going to talk about tetracycline. The mechanism of action of metacycline. Tetracycline is that the tetracycline binds to the mRNA and blocks the attach of the tRNA. The aminoglycosides, they bind to the ribosome, just causing mismatching. But here, they attach to the codon, and the tRNA are not able to bind. Okay, so if the tRNA are not able to bind, the elongation of the peptide chain stops. Okay, in the other case, the peptide chain continues, but it forms wrong protein. But here, the elongation stops, so the protein is not formed. Then, three major modes of resistance. First one, alteration of the drug target. So, again, if my drug attached to my ribosome, the bacteria change the format of the ribosome, for example, by adding methyl groups, and then the antibiotic cannot bind anymore. Another way is by reduced cellular uptake. So the bacteria reduce the number of proteins that it has. Then the number of proteins is gonna, if I have less proteins, I'm gonna uptake less antibiotic. And the last one is the flux pump. So if I have antibiotic inside my cell, bacteria will pump them out by using their efflux pump. Okay, now I'm gonna talk about macrolides. So macrolides also prevent the elongation of the peptide chain, but you doing a different job. This antibiotic prevents the bind of the amino acids. Okay, the tetracycline does not allow the tRNA to bind to the codon, but here the tRNA binds to the codon, brings the amino acid, but the amino acid cannot be bind to each other, so the peptide chain does not elongate. Okay, so amino acid not able to bind. Okay, so my protein is not formed. This is more effective against gram-positive because gram-negative are impermeable to macrolides. What does that mean? It has reduced cell uptake. So gram-negative does not take much macrolides. So this is more effective against gram-positive. There are two major modes of resistance. The first one, again, is by changing the format of my ribosome. So here, my ribosome that's green, each shape it fits perfectly to the antibiotic. But if I change my ribosome, for example, in pink here, my antibiotic cannot bind. Okay, I can change my ribosome by two main ways. First is by methylation. Okay, so I add methyl group to my ribosomes, or I do a mutation in my ribosome. Okay, if I do a mutation in my ribosome, I reduce my macrolides to bind. This was reported in clinical isolates of Streptococcus pneumoniae. Okay, so pneumonia, S. pneumoniae, has been reported to create mutation in their ribosomes. And another one is just the flux pump. So this is abundant in Staphylococcus aureus. So the bacteria just take the antibiotic and pump them out. This is very relevant in cancer treatment. Okay, so if I have this and the person has been treated by chemotherapy drugs, and I have this bacteria will pump out my drugs, so my drugs will not be effective about my cancer treatment. Okay, now I'm gonna talk about fluoroquinolones, but first I'm gonna just give a brief explanation about DNA gyrase and topoisomerase. So, when my DNA has been duplicated, we need to open the double chain. But when I do that, I made a torsion in my DNA that if I don't remove this torsion, my DNA will gonna break down or gonna stop the duplication. Okay, so the enzymes that does that overwind the DNA is the topoisomerase. Okay, so they through my DNA, making that torsion to not happen. Okay, so what fluoroquinolones does? So as I said, topoisomerase is important when I'm duplicating my DNA. Okay, so if I'm duplicating my DNA, I need this enzyme to overwind my DNA. What fluoroquinolone does? It binds to my enzyme topoisomerase. So if I bind to that enzyme, my DNA will not be overwhelmed and my DNA is gonna broke or it's gonna stop the duplication. So bacterial DNA synthesis is inhibited. This has effect against gram-positive and gram-negative. So both of them can be affected by fluoroquinolones. Now, how bacteria got resistance? First, by altering the drug target. So the bacteria creates a mutation in the enzymes DNase and topoisomerase. And then if my enzymes has changed, my antibiotic will not be able to bind them. Another one is just reduce cell uptake. So I decrease the number of antibiotics that I getting. This is most effective in gram-negative, okay, because it has more permeability. And it's not an issue for gram-positive. And another way is the flux pumps. So it's low. Flux pumps is like low levels of resistance, and it may happen mainly with Staphylococcus aureus, Streptococcus pneumoniae, Bacillus species, Mycobacterium tuberculosis, and E. coli. Gonna call here DHFR. How bacteria get resistance? So bacteria, first way, if the antibiotic is destroying my DHFR, bacteria will produce more and more of this enzyme. So if bacteria produce a lot, I will not have a lot of antibiotics to destroy this enzyme. And another way is creating mutation in my enzyme. So bacteria creates a mutation in my DHFR, and then the antibiotic will not be able to attach to that mutated enzyme. This is it. Thank you so much. I hope this helps.